In 2012, Moore et al. Conducted this repeated measures neuroimaging study with adolescents at the ages of 10 and 13 to measure their neural responses to various affective facial displays. The idea behind this research question revolves around the premise that adolescence is a critical developmental period in one's social life where the processing of socio-emotional stimuli becomes increasingly important. We know that hormones start rushing and children's reactions to their peers begins playing as large of a role as ever in the progression of their lives. So, how is their perception of social stimuli changing as they develop through this crucial pubertal period of life? To test this research question, Moore et al. Got 45 children to complete pubertal developmental scale questionnaires, their independent variable, then placed them in functional magnetic resonance imaging machines, once at 10 years old and another at 13. While in the MRI, participants observed pictures of various facial expressions demonstrating different emotions. Their blood oxygenation level responses to the faces were then recorded, which is the dependent variable, and thus Moore et al. Were able to measure their neural responses to affective facial displays in the early stages of puberty in contrast to 3 years later. The researchers found that at age 10, the most active regions responding to these social stimuli were the extrastriate cortex, the thalamus, and the amygdala. However, by age 13, they observed activity in the left temporal pole, ventrolateral prefrontal cortex, and ventromedial prefrontal cortex additionally. Moreover, at 10 years old, the amygdala was only modulated by pubertal development during viewing neutral faces, but by age 13, the amygdala produced significantly greater activation for all 5 of the facial expressions shown. It is also important to note that an interaction between time and pubertal development was discovered due to the 2 different correlations between pubertal development and blood oxygenation level response to emotional versus neutral expressions. At age 10, no significant relationship was observed. However, at 13, pubertal development was positively correlated with activity in the visual cortex, right amygdala, bilateral hippocampus, and temporal pole. This proved to be statistically different than the findings from time 1, implying that neural response to emotional expressions versus neutral ones shows 2 significantly different reactions at these different points in time. So, time is interacting with pubertal development. It seems that by the age of 10, children are primarily only demonstrating a fast evaluation of social stimuli, but by age 13, they're beginning to show an additional slow evaluation to these stimuli that's further integrated in various brain regions. So, what could be done next? A third repeated measure could show us how puberty, and not just age, is associated with ongoing enhanced reactivity to socio-emotional stimuli, as expected in the social reorientation of adolescence. It would also be preferable to measure additional variables such as hormones and physical examinations to define pubertal development. When considering the transition from elementary to middle school, early romantic relationships, and a shift in self-concept, these findings are vital in the context of changing childhood. Ultimately, we gain more insight as to the unruly minds of teenagers, which could eventually help us with our understandings of mental health problems and severe risk-taking behavior.